I'm recording this. Share my screen. Don't think we need that. All right. So we're doing lab 8, identification of gram-positive bacteria. And you should be working on your unknown project. What you should be doing is putting in your unknown notes, your unknown number, and then the results that I sent you on Tuesday. And then look at those results and look at the table of the unknowns. I forget what that's called. I think it's called the unknown list or something like that. And look at that. And then decide what test you want to run next. And then in your unknown notes on the bottom of the page, request that test. Any questions about any of that? You should also be working on your infectious disease paper. Somebody just emailed me, I think it was yesterday, their project. You should have emailed me that a long time ago because I need to approve it and make sure that nobody else is doing it on that project. And then work on that. That is due Saturday, August 14th. So 1 week from today, this Saturday. Any questions? All right, let's go on to, oh, actually, I forgot. We're supposed to do a little bit of lecture.
Dr. Volk,
Yes,
I just logged in. I'm sorry, what was due on the 14th?
the infectious disease project.
Okay, thank you.
I got the headphone on and it's not plugged in, so that wasn't doing very good. Let's go. I forgot, we do a little bit of lecture in the summer. So let me get the lecture here. And get to it. For those of you who don't remember or if you didn't hear, we do a little bit of lecture in the lab because for some reason when they made the summer schedule, they increased the lab schedule, but they didn't increase the lecture schedule. And I need a little bit more time because we only are meeting for 8 weeks in the summer, not 10 weeks. All right, so we talked about DNA. So let's talk about the way DNA or the information in DNA flows. Within a cell, DNA flow, the information in DNA flows from DNA to RNA to protein. And when it flows to DNA, that's transcription. When it flows from RNA to protein, that's translation. This is the central dogma of biology, that information within a cell flows from DNA to RNA to protein. Each of the information is coded in different ways, and we can call that the language that the information is coded. The language in DNA and RNA is in nucleotides, but the nucleotides are slightly different. It's a deoxyribonucleic acid in DNA and a ribonucleic acid nucleotide in RNA. And then the language would be translated into the language of proteins, which would be amino acids. The flow of genetic information, we've talked about it happening within a cell, but the flow of genetic information can also happen between generations of cells or from parent cell to the offspring cells or daughter cells. We're talking about humans, that would be from two parents to one offspring. The information can flow that way. It happens when DNA of the cell replicates, and then the cell goes under cell division, making the two daughter cells, where each daughter cell gets a copy of the chromosome of the parent cell. The information can also flow between cells of the same generation. DNA in this cell could leave the cell and then be incorporated as DNA into this recombinant cell, where this cell gains DNA from another cell. And that is the flow of information between cells of the same generation. Please note that I've altered the picture a little bit because I think it's a little more clear that the DNA leaves this cell, and it can be naked DNA out in the environment, and then can be incorporated into this cell, the recombinant cell, taking up the DNA. Any questions about any of that? Sorry, I had a frog in my throat. All right, within a cell, the information can flow, and we call this the vertical flow of the information in DNA, where the information flows from the DNA. Language is DNA nucleotides. There's 4 letters in the alphabet, meaning DNA is made up of 4 nucleotides. That information flows to the RNA, and the language is nucleotides. It's a little different. It's going to be a RNA nucleotide, ribonucleotide, but there's only 4 of them. And then the information flows to proteins. The information is in the language of the amino acids, and there's 20 different amino acids that make up the protein. Now, in comparison, English has 26 letters in their alphabet. Those of you who know Spanish, I believe there's 28 letters in the Spanish language. So while the information is in the DNA, we can say the information is coded as, that is a dog. Any question about any of that? Now that's transcribed into the information in RNA, and that information would be similar, a little different accent. That is a dog. And then that information will be translated into the protein, and the information will be different. Quello è un cane, meaning the same, that is a dog, but in a different language. Any question about any of that? If not, let's talk about we've talked about how information flows within a cell. Let's talk about the vertical flow of information. The vertical flow of information happens when the DNA replicates and then goes to the 2 daughter cells. How that happens, let me get this line bigger. Is that the 2 strands of DNA separate, and then 1 strand is used as a template for building a new strand of DNA. So the DNA polymerase will bind to this strand, C and A here, and then put the T nucleotide there. I guess we should do it on this one. So it sees T here, it puts A there. And then it'll move down. You'll see a G here. And so it'll put a C nucleotide to the new strand. And then it'll see on the template a C strand, a C nucleotide. So we'll put a G strand. And the DNA polymerase just goes down the strand, reading it and putting the complementary base pair in position for the new strand of DNA. Any question about any of that? So there are only 4 nucleotides added in DNA, and that's A, G, C, and T. We say that DNA replication is semi-conservative because whenever the DNA replicates, one cell will have 1 strand of the original DNA and 1 strand will be new DNA, and the same with the other cell. So that's semi-conservative. Come on. Semi-conservative replication. The original strand would be 2 black strands. 1 strand will be used as the template to make the new strand, which will be white. And then this will be going into 1 cell where it'll have 1 strand of the original DNA and 1 strand of new DNA. That's the semi-conservative model of DNA replication. The conservative model would be when the DNA replicates, 1 strand would get, or 1 cell would get 2 strands of black DNA, meaning the original strand, and then the other cell would get 2 strands of white DNA or the new strand of DNA. And that model doesn't happen with DNA replication. Don't ask me what the liberal model would be for DNA replication. I don't know. Any questions? I've already talked about that. How it gets added is the DNA polymerase adds the correct nucleotide, and it's going to be a nucleotide triphosphate, meaning there'll be 3 phosphate groups added onto it. And for adenine, if it's adding adenine, that would be ATP it's adding. Okay, but 2 of the phosphates come off, and then we have the phosphate linking to the ribose, and that's a covalent bond. And the phosphate, or the sugar from the ribose to the phosphate to the ribose, is the sugar phosphate backbone, which holds 1 strand of DNA together. The nucleotides do not hold a single strand of DNA together. What the nucleotides do is they hydrogen bond to the 2nd strand of DNA. And this is not a covalent bond, it's hydrogen bonded. Any question about that? All right, let's watch a little video on DNA replication. And get it to open. Are you seeing this?
Yes.
Okay, good.
DNA is a molecule made up of 2 strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The 2 strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand. And wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
All right, any questions? Oh, go back. Oh, too late. All right, the first enzyme you need to know about in DNA replication is helicase. It makes the double strand of DNA single-stranded, so it opens up the DNA. The second enzyme you need to know is, I just saw it here, DNA polymerase. It puts the nucleotides on the growing chain of the new DNA strand. So it adds the nucleotides. And how it adds it, it reads on the template strand and then puts the complementary nucleotide on the new strand. So DNA polymerase is the second enzyme you need to know. The third enzyme that you need to know is ligase. Let's see where that is. There are other enzymes, but those are the three main ones. There it is. DNA ligase. On the the leading strand, the ligase only needs to work in two places. Where the strand starts, so let's say there was a strand here and then the RNA or DNA polymerase bound here and then made this new strand of DNA. Well, ligase will need to link the new strand of DNA to the starting position, this the old strand. And then where the new strand stops, the polymerase stops, and let's say falls off, and let's say there's another strand of DNA there. The ligase will need to link that together. So in the leading strand of DNA, the ligase doesn't work more than about twice. Where the DNA polymerase starts and where it ends. On the lagging strand, however, let me see if I can point that out. Well, it doesn't have it shown very well. Let me see if I've got it better here. Here it is better. On the lagging strand shown here, we have several Okazaki fragments. And each Okazaki fragment has to be ligated with the fragment in front of it and the fragment behind it. And so each Okazaki fragment, ligase will act 2 times, and there will be many Okazaki fragments because DNA has to be made in this direction, the 5' to 3' direction, but the DNA polymerase is moving that way, and that's the wrong way to make the DNA. And so the DNA is made in short segments, I don't know, not backwards, but in this direction. And that we call Okazaki fragments after Dr. Okazaki, who discovered it. Any question about any of that? I think that's it for the video. There are some other enzymes, but I'm not going to ask you about them, like primase, hexanuclease. All right, do you all understand the difference between the leading strand and the lagging strand? No comments, so let's move on. New DNA is made in the 5' to 3' direction. It's initiated by an RNA primer. The leading strand is synthesized continuously. The lagging strand is synthesized discontinuously in little chunks that we call Okazaki fragments. And I could be butchering his name. I'm not Japanese. And I don't speak Japanese. RNA primers are removed, and then the Okazaki fragments, as well as the breaks in the leading strand, are ligated together with DNA ligase. So this is the only enzyme. Well, there's DNA polymerase, DNA ligase, and helicase starts it by making the double-stranded, single-stranded. The enzyme unwinds the parent double strand. Helix is number 1. It's not mentioned helicase, but that's the name of the enzyme. Any questions about DNA replication? If not, I think I will end here. We only have 4 minutes. That won't be enough to get through transcription. So I'm going to stop here and continue with the lab. So we're doing lab 8. And I don't need my thumb drive for that. Lab 8 will have a number of case studies, 8 of them, as well as a laboratory exercise, which please put your answers on the exercise of the worksheet. And there's also a lab module. That's the one I want to open. All right, lab 8, the identification of gram-positive organisms. We're going to look at gram-positive cocci, the Staphylococcus and the Streptococcus. We're also going to look at a few gram-positive bacilli, Enterococcus. That's not a bacilli, that's a coccus. That shouldn't be there. And Listeria, which is a gram-positive bacilli. Let me move that over, because that's in the wrong place. There's some video clips you'll be watching. The dichotomous key, which I should probably show you that. It'll take me a while to get to, so let me get to.
Dr. Volpe,
Huh?
I think the lab module's not on Canvas. I think it's the exercise posted twice.
Uh-oh. All right, let me take a look. I do need to get there. I was hopefully going to do that while I was talking. Why am I not seeing my key? I doubt that I dropped everything. For some reason it's hidden, and I'm not sure how to get it. So let me, gosh, this is, I'm having trouble here. Just a minute. For heaven's sakes. You might have to, there it is. Put that up. That up. They're saying that's the worksheet.
When I click the module, it looks like the worksheet.
Yeah, there's a problem there. All right, let me, see if I've got it. We're on module 8, right?
Yeah.
Probably this one. Let me look. Yep. Maybe at the bottom. All right. Other than it's at the bottom. It should be up front. There's the module. Thank you for pointing that out to me. One more. Right there. Ooh. Now there's two of them here. That's bad. I think this is the one we want.
I think so too.
Yeah, it's the module there, and that one's that one. So let's close that down and remove this one. All right, you might have to refresh your page, but that should now work. Lab module 8, which I've got there, so that works. And I don't want to talk from there. But you'll note, let me back up a bit there, that we have a worksheet. Please put your answers on the worksheet. And you have 8 case studies. You have to link on 8 different PDF files. That's the way the department made it. And I've never tried to put them all in one file because they're PDF files, so they're hard to work with. So take a look at the case studies, read it. It gives you information on a patient who has an illness. Not going to show well in Canvas, but spider bite, gram stain, growth on blood agar plate, some tests that were done on the bacteria, more tests, more tests, and then it's asking you what bacteria was isolated, what you want to do to isolate the bacteria is take a look at the dichotomous keys and use it to isolate your bacteria based on what you know about the bacteria, what you know about the infection, and what the test results are. So the gram stain was done, check whether it's gram cocci. No, check whether it's gram positive or gram negative. Obviously it'll be gram positive for this lab. Then look and see, is it a cocci or a bacilli? If it's a cocci, then you run the catalase test. If it's catalase positive, it's staphylococcus. If it's catalase negative, it's streptococcus or enterococcus. If it's staphylococcus, you can do the coagulase test, in which case if it's positive and coagulase, the Staphylococcus aureus. If it's coagulase negative, it's another staph. And then after that, you can do the novabiocin test to separate out Staphylococcus epidermidis from Staphylococcus sapropsychus. Okay, so you need to use this dichotomous key to figure out what bacteria you're using, you're finding. Please only use this dichotomous key for this lab, especially your unknown project. You cannot use this dichotomous key for your unknown project, meaning this has species that we do not have in the unknowns list. And then we might have some species that they don't have in the dichotomous key. And this is the unknowns list, giving you the number of bacteria you have to consider, which are on the y-axis here, and then the possible tests you can run are on the x-axis. All right, any question about any of that? If not, let's go on to talking about the lab module. Unfortunately, I don't have an icon at the bottom. There it is. So there's some reading you should do before you do the lab. And it's not here. That's weird. Let me see if I can find it. I need my icon. Oh, where the heck is it? All right. We're going to skip that. Ah, jeez. My computer isn't working right, so I'm having a hard time. Let me stop sharing the screen, and maybe that will allow me. Yeah, that did work. Let me... Oop, that's the wrong button. We're going to close that down. Now, how do I do this? I'm trying to share the screen, but not... There we go. Got it. That was tricky because I didn't have any buttons down here. I didn't know what to do with no buttons. Upon successful completion of this lab, students should be able to understand the principles and how to interpret the results for the CAMP test, the Bialis-Sculen test, the SF-Broth test, and the Opto-Chen sensitivity test, and be able to use a dichotomous key to solve case studies involving gram-positive bacterial infections. Any question about your learning objectives? When specimens are received in a microbiology lab for routine bacterial cultures, they're inoculated on culture media that will facilitate the growth of common pathogens and placed in physical environments that support their growth. Microbiologists do use the colony morphologies and the gram stain to provide important information about the bacteria that they isolate. However, you cannot identify the species on that alone. Further biochemical tests have to be done before you'll get the information to determine what species you have. In this lab, we're going to look at the CAMP test, the Bialis-Sculen test, the SF-Broth test, and the Opto-Chen sensitivity test. Any question about what we're doing? CAMP is an acronym for the three microbiologists who invented the test. And you don't really need to know Christy Atkins and Munch Peterson, but it's CAMP. And they first described an amplified hemolytic effect that some beta hemolytic organisms have when they are growing physically near Staphylococcus aureus. The single streak of beta hemolytic Staphylococcus aureus is put down the middle of a blood agar plate. And then perpendicular to that streak of Staphylococcus, we streak the other bacteria, which will be the bacteria we're testing or the unknown organism. And then you look to see if there's amplified hemolysis. that doesn't look good. Oh, there it is. Amplified hemolysis near the Staphylococcus aureus. Streptococcus pyogenes, when you grow it near Staphylococcus aureus, it's negative in the CAMP test. There's no added hemolysis near Staphylococcus aureus. Listeria, when we streak it this way, near the Staphylococcus aureus, we have slight positive CAMP test, meaning we have slight added hemolysis in this region compared to that region. With Streptococcus agalactia, we have tremendous hemolysis. So this is positive CAMP, where we make a single streak there and almost no hemolysis on the smear until you get near the Staphylococcus aureus, in which case the hemolysis takes on almost like an arrowhead shape. And there you can see the streak of the bacteria, and that white area is the amplified hemolysis we have around Staphylococcus aureus. You can see the streak of Staphylococcus aureus right here, and then this clear region is the hemolysis from the Staphylococcus aureus. Any question about the CAMP test? All right, biolisculin hydrolysis test. Biolisculin is grown on a slant, and it's used to differentiate the Enterococcus from the Streptococcus. If the organism hydrolyzes asculin, they will produce asculatin, which will appear black or very dark on the biolisculin test. So that's biolisculin positive, where asculatin is made. And this is biolisculin negative. Streptococcus species or biolisculin negative, Enterococcus species or biolisculin positive. All right, any questions about biolisculin? SF broth is Streptococcus faecalis broth test. It's used to differentiate between Enterococcus faecalis and Streptococcus other species. Enterococcus faecalis, the old name for it was Streptococcus faecalis, but they realized it's not a member of the Streptococcus family when they sequenced the DNA, so they now call it Enterococcus. But this SF broth is for distinguishing between those two different species or family. If the organism grows in the broth, it will ferment the sugar glucose, producing a yellow color. And that's from the accumulation of acids with glucose fermentation. If the organism does not ferment the sugar glucose, or it does not grow in the broth, it'll be negative, and the color will remain purple. Streptococcus faecalis will be positive. Excuse me, that's the old name. Enterococcus faecalis will be positive, and the Streptococcus species will be negative. The Optogen sensitivity test is testing the bacteria for sensitivity to the antibiotics Optogen. And it is used to differentiate Streptococcus pneumonia from other alpha hemolytic streptococci. So when you streak out bacteria on a blood agar plate, if you have alpha hemolysis, which that's not shown very good, blow it up. There you can see darkening around the bacteria. It's most prevalent right in this region, actually, the darkening of the plate. And then we take this bacteria and put it on a plate, and then put the Optogen on. It's on the paper disc. And if there's, this is the bacteria, the darkening of the plate, making it, the bacteria look dark from partial hemolysis of the blood. There's no sensitivity to Optogen here. The bacteria grows up to the disc. So this would be Optogen resistant bacteria. So it's not Streptococcus pneumonia. If on the other hand, the bacteria is sensitive to the Optogen disc, and you have a zone of inhibition greater than or equal to 14 millimeters, we say that the bacteria is sensitive to Optogen, and Streptococcus pneumonia is sensitive to Optogen. Now, I should mention that to be resistant, you don't have to have no zone of inhibition. You can have a zone of inhibition that's less than 14 millimeters, and we still say that that bacteria is resistant to Optogen. Any question about any of that? All right, so that's the Optogen sensitivity test. Once you ran these different tests, you then get your data and your results and look it up on a dichotomous key. And then determine what the species will be from your dichotomous key. If you don't know how to use a dichotomous key, we've got a how to use a dichotomous key and using dichotomous logic. Dichotomous logic is where one test will give you two possibilities, like the gram stain. You can have gram positive and gram negative. So that's a dichotomous test. If it's gram positive, following dichotomous logic, you do another test after that, which separates down into two. To follow dichotomous logic, you do one test at a time, and that test will separate the data into two possibilities. And you look at what your results are, figure out what it is, and then look at the chart of the unknowns, and then decide your next test. So that's following dichotomous logic. Gram stain, this I should have came here, gram positive, gram negative. You can then do the shape of the bacteria, bacillus or cocci. If it's cocci, you can do the catalase test. Catalase positive, it'll be Staphylococcus. Catalase negative, Streptococcus or Enterococcus. If it's catalase positive, you can then do the coagulase test for positive and negative. If it's positive, it's Staphylococcus aureus. If it's not positive, meaning coagulase negative, it's some other Staphylococcus species, and you could do the Nova Bayesian test for separating out the two species that are coagulase negative. Do not use this key for your unknown project, and do not use this key for Lab 8 as well. Okay, any question about any of that? All right. So in doing the lab, you're not physically going to be performing any of these activities. However, you will be responsible for knowing how to perform and interpret these tests. View the following video clips. We have one on the SF broth and the bile esculin test, and then another video on the CAMP test. Review how to interpret the following test by completing this chart. We'll do that in the worksheet. Let me show you that. So fill in the worksheet for the positive results and the negative results. Some of these, like the coagulase test, the CAMPS test, the biolosculum, came from this lab module. So you'll get your answers in this lab module. But some of them came from a previous lab module. So you have to look for some of the answers in an earlier lab module. Like when you did the Schaefer-Fulton stain for endospores, I think that was in lab module 7, but I might have the wrong. Yeah, I'm pretty sure that's throat and urine cultures for lab module 7. All right. I'm just a little confused here. There we go. So some of them will come from lab module 7. There may be 1 or 2 that come from an earlier lab module. But just, you can get all of your answers from the lab module. And if you're stumped, you can also get your answers from online. There's a video for how to develop a dichotomous key. If you're comfortable with developing a dichotomous key, then you don't need to view this, but it's there in case you need it. This dichotomous key you do not use in this lab module or for your unknown project. It's just to help you work with a dichotomous key. There is a dichotomous key on Canvas that what you'll do is use it for studying these bacteria, which are the bacteria that can cause the case studies. And there are 8 case studies. Let me see if I can show you that. Click on the links for the different case studies, read about the case studies, and then you have to give the species name that is causing the infection for case study number 1. Don't give me the disease name, give the species name that's causing the infection. Any question about lab module 8?
Yes.
Yeah,
Do you want us to put how we got the species or just the species name for the?
just the species name.
Great, thank you.
Okay. All right. No, you don't need to put out your dichotomous key for how you did that. That would be a lot of work. You'll notice that this lab has a number of case studies, and each one of them is a bit of work. So this lab will take you longer to do than most of the other labs. Okay. All right, get started on it. I will be here until 8:20 to answer any questions. And try and answer your questions before that, because I doubt that students will stick around to 8:20. But I'll be here until the last student logs off or until 8:20. I will be here at least until 8 o'clock. All right. Okay.